of your DNA video. Our last key point is going to be replication, transcription, and translation. So what replication is, it's called DNA replication, and this is where DNA makes a copy of itself. And we talked a little bit about this. This happens during the S phase of that cell cycle. So remember cell cycle G1S, G2M, one cell comes in, two identical cells come out. Well, during that S phase of the cell cycle, the DNA replicates or it makes a copy of itself. And this is how it happens. So here's my structure of DNA, and you can see my nucleotides. So here's an A, it's paired with a T, a C is paired with a G, G to C, and so on and so on. But what happens is there's a special protein, an enzyme that called DNA helicase, and it comes through here and it unzips these genes. It separates them, so they're not base paired together anymore. And because they're not base paired together anymore, that allows other nucleotides to come in here and base pair. So another type of enzyme will come through here and it will add a T to the A, and a G to the C, and a C to this G, and a T to this A, and an A to this T. And then it will do it on the other side also. So it'll add an A to the T, and a C to the G, and a G to the C. And what that does is that creates two identical strands of DNA. Here's my original strand in green, A to T. Well, my new strand is also A to T, and this other one is also A to T. But half of it was my original DNA, and then half of it is new DNA. And I have two identical strands of DNA now. And that's the simple process of DNA replication. And this right here is going to be called semi-conservative. It's not completely new. It's not old. It's partly old DNA and partly new DNA. So here's those steps. This enzyme DNA helicase will unwind and unzip my DNA. Once they're not base paired anymore, another enzyme called DNA polymerase will come through and it will start base pairing A to T and C to G. And the result is going to be two new strands of DNA, or two identical strands of DNA. And then, through the cell cycle, these will be separated into two identical cells. All right, so that's DNA replication. Now the next thing is going to be transcription. And remember, DNA makes RNA, and that is what transcription is. Transcription is when DNA makes a strand of RNA, and we're going to call it messenger RNA or mRNA. And it's kind of the same process as that replication. So in the blue here, that's my DNA. And if you look closely, you can see, the T to A and C to G. But there's going to be an enzyme. It's a different enzyme. It's called DNA polymerase. It's going to come through and it's going to unzip my genes. So it's going to pull them apart. And it's going to expose these nitrogenous bases. And then one of these sides is going to build that RNA. So if I have, a C, then it's going to pair with a G, I'm going to add a C, and A, I'm going to add a U, because this is RNA, and then G to C, A to U. And here I have a strand of RNA, so my red strand here, that is my messenger RNA. And this is going to be what leaves the nucleus and goes and builds that protein. So DNA makes RNA. That is called transcription. So here's my steps to it. DNA unwinds. New RNA or nucleotides are combined with that DNA. They base pair. So if the DNA is an A, I'll pair it with a U. If it's a T, I'll pair it with an A, a C to G, and a G to C. And then I'm going to have a strand of messenger RNA. Now that messenger RNA is going to leave the nucleus, and then it's going to find a ribosome. So here's my ribosome, the little blue thing in here. And it is going to make a protein. And that process is called translation. So when RNA makes protein, it's called translation. And here's my strand of RNA, that red strand here. And it's just going to feed through this ribosome, which is that protein factory, and it's going to build a protein. And some important things in this picture is this right here. It's called a codon. And what a codon is, it is 3 nitrogenous bases. So in this case, it's a U, an A, and a G. And they're going to code for one monomer, or one building block of a protein. So every 3 nucleotides is going to code for one amino acid, which is the monomers for my proteins, my polypeptides. So here's one amino acid, here's another amino acid, another one, another one, another one, another one, and they're just adding these together. This is what your ribosome does. It puts these amino acids together to build my protein. So that is translation, when I take this RNA and I make protein. Now, there's different types of RNA. The one that we care about is called messenger RNA, or mRNA. That's what's made through transcription. There's another type of RNA called tRNA, or T for transfer RNA. And what these do is they just transfer the amino acids to the ribosome, and they kind of base pair with your messenger RNA, and that's how they know what amino acid to bring to the ribosome. All right, so here's my steps to translation. My messenger RNA finds a ribosome. That ribosome uses the codon and the RNA to start combining amino acids. So once again, a codon is a sequence of 3 nucleotides, and it codes for one amino acid. And those amino acids are the building blocks, the monomers for a protein. Now, if I put these amino acids in a different order, the protein will be different, and it will do a different job. So remember, DNA makes RNA, RNA makes protein. A gene is just a section of DNA that is going to build a protein. So every single protein in your body is going to be different, and it has one job. So here's that whole process. So here in the gray, I have my DNA. I'm going to unwind my DNA, and I'm going to start base pairing, and I'm going to build RNA. I might have said RNA there, I meant DNA. So I'm going to unwind DNA, I'm going to base pair, and I'm going to build RNA. And then that RNA is going to leave the nucleus. It's going to find a ribosome. So this white thing here is a ribosome. And every 3 of these nucleotides is going to code for one of these amino acids. So all these little circles here, those are the amino acids for my protein. And then this big long line of circles, that is my polymer, or my polypeptide, or we like to call it a protein. So that is replication, transcription, and translation. If you have trouble remembering the order these go in, it's alphabetical. So replication, DNA to DNA, comes first, right? That's during the cell cycle. Then transcription, DNA to RNA, comes before translation, which is RNA to protein. And it's alphabetical. So if you look here, C comes before L, right? So transcription will always come before translation. So transcription is DNA to RNA, and translation is RNA to protein. All right, so that is everything for your nucleic acid C notes. So after you're done with your escape room, make sure you type your C notes up into the appropriate section in your notebook. This section is called What is DNA and Why is it Important? There is a link for it in the Biomolecules homepage. Like your other ones, take a screenshot of your typed C notes and then either email them to me, and then make sure you finish your escape room for a grade.